Now, acid-base properties of amino acids. In previous video, we have seen the meaning of amino acids and different properties related to the amino acids, their classification also we have seen, and even we know the structures and abbreviation of 20 amino acids which occur in nature. Now see, amino acids are actually having both NH2 as well as COOH group. If NH2 group is there, there is a lone pair over the nitrogen and it makes this amino acid as a base. So it is a base because of NH2 group. And because of this COOH group, it can give proton, this proton. And because of this, it is acid. So both acids and base, both properties are there on the amino acid. But actually, amino acids are existing as a salt because there is intramolecular transfer of proton from the COOH group to the NH2 group and they are converted into the salt. So because of this salt, they are always having melting point, high melting point, and they are highly soluble in water. What happened here? In amino acid, this proton, since acidic group and the basic groups are very close and they are present in the same molecule, H is transferred from COH group to the NH2 and this H is now forming bond with this nitrogen by using the lone pair. So when this lone pair is vanished, it will produce a positive charge. And when this proton is lost from this COH group, it will produce a negative charge. So net charge on the amino acid is 0 or the neutral. So this ion is called actually positive and negative charge. So is called zwitterion. So this zwitterion is neutral because of positive and negative charges, cancellation of positive and negative. So this is formed, zwitterion is formed because of the proton transfer and this is called salt. This is called salt. Now, when we change the pH of the amino acid, when the amino acid is taken in solution and if you change the pH of the amino acid, it can exist in 3 different forms. And these 3 different forms are actually the function of pH of that solution. I have taken here one example of alanine where R is replaced. This is a general form. R is replaced with CH3. This amino acid is called alanine. So actually, when we maintain the pH of that solution as a neutral pH, where the pH is approximately 7, which is neutral, this amino acid exists as a neutral zwitterion. So this is zwitterion when the pH is around 7. But in the same solution, if you add strong acid, this zwitterion is converted to this form where the pH is equal to 2 or less than 2 in presence of strong acid. So what happens there? That strong acid provides proton to this carboxylic group and this negative charge is now gone. But this proton will remain there on the nitrogen because in the reaction medium there are a lot of protons available. So when the pH is less than 7, the negative charge is gone and the net charge on the amino acid is positive. So in presence of strong acid or when the pH is less than 7, which is equal to 2 in case of alanine, or less than 2, this amino acid is having net charge positive because of this. And when strong base is added, so you know when base is added, pH is increasing, pH is more than 7. So in case of alanine, if pH is equal to 10 or more than 10, so when base is added, this proton is removed, this proton is removed. So when this proton is removed, the positive charge will be gone, and when this proton is removed, a negative charge is developed. So net charge on the amino acid is negative. So in presence of strong base, the amino acid becomes negative, and in presence of strong acid, amino acid becomes positive. And when the pH is neutral, when the pH is around 7, the amino acid becomes neutral. There is no net charge. There is no negative or positive charge, net negative or net positive charge. So in this way, amino acid can exist in 3 different forms, and which is the function of the pH. When the solution is, we are going to, if you change the solution's pH, it is converted to neutral to positive, or positive to negative, or anywhere. We can change the charge on the amino acid when we change the pH. Now when we discuss isoelectric point, we must know the structure of zwitterion. So that zwitterion is NH3 plus COO minus. This is H and this is R. So here this is NH3, and when we add acid, when we add strong acid, there is a proton attached here on COOH group. So we can say this is a protonated form of amino acid. So when the protonated amino acid, we consider there are actually, we have 2 protons, this proton and 1 proton from here to remove. And because of this, each amino acid have 2 pKa value. Each amino acid have 2 pKa values. For example, pKa value of carboxy proton in case of alanine, when we replace R with CH3, is 2.35, and the pKa value of ammonium proton is 9.87. This is for alanine. So there are 20 amino acids, and all 20 amino acids having different pKa values. Each amino acid having 2 pKa values, because in protonated amino acids we have 2 protons to remove. Now the pH at which amino acid exists primarily in its neutral form. In its neutral form means what? In its zwitterion is called isoelectric point. So when it is in neutral form. Neutral form means when there is no H here, there is negative charge, and here it is positive charge. So net charge is 0. So neutral ion. So that exists for certain pH, and that pH is called isoelectric point. So each amino acid having its own isoelectric point, and it is represented by Pi. This abbreviation is used. So to form a neutral molecule, to form a neutral amino acid, what pH is required is called its isoelectric point. Generally, that isoelectric point is nothing but the average of both pKa. So pKa plus pKa2, pKa1 and pKa2 upon 2. So this is given here. Isoelectric point is equal to pKa of COOH plus pKa of NH3 plus divided by 2. So this, when we take the average of this, we get isoelectric point. For alanine, we have 2 values. One is for COH group, it is 2.35, it's pKa, and for NH3 ammonium proton, it has 9.87. So when we take the average of these values, that comes 6.12. So 6.12 is the pH where alanine exists in neutral form. So this is the isoelectric point of alanine. Thank you.